Hello all, welcome to my channel. In this video, we are going to discuss about search for genetic material, and as we know, DNA is the genetic material. So when, how it concluded that the controversy end that DNA is the genetic material. It is concluded, it is put a stone by Griffith experiment. So let's discuss about Griffith experiment for search of genetic material. The search for genetic material started during the mid-19th century. The principle of inheritance was discovered by Mendel based on Mendel's investigation that some factors are transferred from one generation to another. It took a long time for the acceptance of DNA as the genetic information. Let's go through a brief account on the discovery of a genetic material and Griffith experiment. Griffith conducted an experiment in 1928 where he took the bacteria Streptococcus pneumoniae for his experiment, and this bacteria caused pneumonia. During the experiment, Griffith cultured Streptococcus pneumoniae bacteria which showed two patterns of growth. Here is the two pattern of growth in the photographic image. One is rough strain or R strain, another is smooth strain. Smooth or S strain is shiny mucous polysaccharide coat. These are the coat, double coated layer. Polysaccharide coating is the labeling. Polysaccharide coating, it have the outer covering or coating which is nothing but a polysaccharide coating. Resistance to immune system and virulent means poisonous strain. Next is R strain. It doesn't have any polysaccharide coating. It lacks its coat, destroyed by immune system of the host, and non-virulent. Immunity power of the host body can destroy these R strain bacteria. So it is non-virulent or non-poisonous. Here S strain or smooth strain which is resistant to immune system. It can beat the immunity of the host body. So let us discuss what Griffith do in his experiment. First, Griffith took S strain and injected into the mouse, and he found that the mouse is died by pneumonia. As we discussed in our last slide that S strain is resistant to immune system and it is virulent. So when this virulent strain bacteria injected to the healthy mouse, the mouse is died due to pneumonia. In the second step, Griffith inject another healthy mouse with a virulent strain or rough strain, as it is bit by the immune system. So here the mice is not affected by pneumonia and it is live. In third step, the S strain or the virulent strain was heat killed, and this virulent heat killed strain injected into the mouse, and the result, the mouse is live. And in his last step, he mixed the heat killed S strain with R strain and injected into the mouse, and he found that the mouse is died. Then he took the blood sample of that dead mice, and in the blood sample he identified the bacteria pneumonia. So he concluded that the mouse is died because of the bacteria pneumonia. From the experiment, he concluded that something caused bacteria to change from one type to another. That is, the raw strain becomes S strain, smooth strain, or the virulent strain becomes the avirulent strain becomes the virulent. So he concluded that something is transferred from the heat killed smooth strain to the raw strain, for which the raw strain is behave as virulent, and he named that as transforming principle. Something is a transfer, but what is transfer? The transforming principle was still unknown for Griffith, but he only assumed that this transforming principle is nothing but somehow the genetic material. But what exactly the genetic material? What is transfer? He still unknown about that genetic material. After Griffith, three scientists, that is Avery, MacLeod and McCarthy, start the Griffith experiment again, as Griffith experiment was the turning point toward the discovery of a hereditary material. These three scientists start the experiment where Griffith left. So, Avery, MacLeod and McCarthy continued the Griffith experiment in search of a biological nature of a hereditary material. Their discovery revised the concept of a protein as genetic material to DNA as genetic material. Avery and his team extracted and purified protein, DNA, RNA and other biomolecules from that heat-killed strained bacteria. Our point is at that heat killed. From that heat killed S strain, something is transferred to R strain. That's why R strain behave as virulent or poisonous. So something is there in the heat killed S strain bacteria. So Avery and his team extracted and purified protein, DNA and other biomolecules from that heat killed strain, and each biomolecule he extracted separately from that heat killed strain, injected separately to individual mice. They discovered that DNA is the genetic material and it is alone responsible for the transformation of a R strain. So, here is the search end for the genetic material. And it was concluded by Avery, MacLeod and McCarthy that DNA is the genetic material. Here we finish with search for genetic material, and there is answer, DNA. Search for genetic material, that is DNA. Here we finish. If you have any query, please comment in the comment box. If you need any changes, any suggestion, you may also comment. And please like, share and subscribe my channel. Thank you.